This protocol shows the production of high-quality plasma retinol binding protein that is used to study the activities of retinol binding protein receptor in real time. This is achieved by first expressing and purifying a large quantity of retinol binding protein from bacteria. The purified retinol binding protein is completely denatured and slowly refolded in the presence of its ligand retinol. Then, the correctly refolded retinol binding protein is purified using HPLC to remove incorrectly folded protein and bacterial contaminants. Once high-quality retinol binding protein is obtained, it can be used to study the activities of its membrane receptor in catalyzing retinol release, retinol loading, or retinol transport to cellular retinol binding protein 1 in real time.
Today, I will demonstrate our improved protocol to refold and purify retinol binding protein producing bacteria. This technique can produce high-quality retinol binding protein, which is essential for functional assays.
I will demonstrate the newly developed techniques to study the receptor for retinol binding protein. The main advantage of these techniques over existing methods, like radioactivity-based or HPLC-based techniques, is that it allows you to monitor the progress of multiple reactions in real time.
The most difficult part of this procedure is RBP refolding, which can both determine quality